Hello, thank you all for joining us. We will begin the NOAA Omics Seminar in 5 minutes. Hello, everyone. Thank you all so much for joining. We're going to begin our seminars promptly at noon. All right, we're going to go ahead and get started. Hello, everyone. Thank you all so much for joining the webinar today. Welcome to the NOAA Omics Seminar Series. I am your host, Catherine Egan. I am the NOAA OAR Omics Coordinator, and I also sit in NOAA Ocean Exploration. I also participate on the NOAA Omics Working Group as a subcommittee co-lead. Omics, which is a suite of tools used to analyze DNA, RNA, proteins, and metabolites, has become a large priority here at NOAA in the last several years. We established this seminar series in an effort to increase transparency and collaboration and highlight the incredible omics-related research currently underway both within and outside of NOAA. The seminars from this series are all made available on YouTube and posted up on our omics website a few weeks after the initial presentation. I'm going to drop a link to that in the chat right now if you'd like to look at our past seminars. You can also reach out to me via our NOAA Omics email account if you have any questions about the omics portfolio here at NOAA. As participants, you all are muted, but feel free to type questions into the chat box throughout both presentations. At the end of each presentation, I will read the questions to our presenters for their answers. So with that, we have two presentations today. The first presentation is Combating the Stony Coral Tissue Loss Disease Outbreak Using Omics, presented by Dr. Michael Sutton. So Michael, while I am going to read your bio, I'm going to give you permission to share your screen. So Dr. Michael Studivan is the lead for disease and omics projects within the AOML Coral program and is interested in using omics approaches to better understand coral connectivity and resilience to stress. Prior to his current position, he received his PhD and completed a postdoc with Dr. Joshua Voss at Florida Atlantic University as part of the NOAA Cooperative Institute for Ocean Exploration, Research, and Technology. His previous research used population genetics and transcriptomics approaches to understand the connectivity and plasticity of mesophotic corals that are between 30 and 150 meters in the Gulf of Mexico and Caribbean, contributing to the Flower Garden Bank's National Marine Sanctuary expansion. Michael is also an avid technical diver and has over 10 years of filmmaking experience as a means to enhance science communication. Thank you so much, Michael, for presenting today. I will now pass it off to you.
Thanks, Catherine. Can you hear me okay?
Yep, we can hear you great.
Great, and it looks like my slides are coming through, so I will begin. As Catherine said, I am the Coral Omics lead at the NOAA OML Lab in Miami. I'm also affiliated with the University of Miami's Cooperative Institute for Marine and Atmospheric Studies. And so before I begin today, I just want to give a shout out as the projects that I'll be presenting on are highly collaborative and have been successful, particularly through the efforts of graduate students and early career researchers. So it's a great team to work with, and I'm proud to present the results of some of our recent work. For those of you who might not be familiar, coral reefs throughout the Caribbean are currently experiencing a devastating coral disease outbreak known as Stony Coral Tissue Loss Disease. It first appeared off of Miami, Florida in 2014 and has since spread throughout the entirety of Florida's coral reef and has now spread to numerous territories throughout the Caribbean. What makes this disease so threatening is that it affects nearly half of all Caribbean reef building species. At the last count, I believe it was 24 species. And there are estimates that it has killed millions of coral colonies throughout the endemic zone. AOML is one of the leaders in the federal response to this disease outbreak, and our group specifically focuses on three research priorities. First, to test transmission sources in an ex situ manner. Second, to identify putative pathogens through omics approaches, as we'll see shortly. And third, to evaluate potential treatment approaches. Now, our research is broadly designed to meet research priorities as well as to have direct applications to management initiatives. And so we have three spatial levels that I'll be discussing today. The first is at the reef level, where I'll be describing one of our recent projects that assessed SCTLD transmission through reef sediments. At the region level, we've also recently conducted an experiment to address the potential for disease transmission through simulated ballast water and the treatment of diseased exposed water. And then finally, at the coral level, I'll be describing some of our recent work looking at the success of disease intervention using antibiotics in the field and addressing whether there are any potential ramifications for long-term coral health. Jumping right in, I'll be starting with the Reef Sediments study here. And throughout Florida's range, there have been an increasing number of studies and hydrodynamic models that suggest that water is the primary transmission source for this disease. But there's been additional limited evidence that sediments may be contributing to the spread as a source or as a disease reservoir. In Florida, there are a lot of coastal construction activities like port dredging and beach renourishment that may have contributed to the spread of the disease throughout all of our coral reefs in nearshore environments. These activities have the potential to transport large amounts of sediments and water from inshore to offshore regions. There's been some work that have found disease indicator microbes in sediments surrounding diseased coral colonies. And so we set out to test whether reef sediments can transmit this disease without direct contact between diseased and healthy corals. We started by designing a novel disease transmission apparatus at our AOML and CMIS wet lab facility. What makes this system unique is that we have split up each individual coral replicate into its own independent vessel. So this allows us to have statistically independent disease challenges, and each coral is supported by its own seawater source. So this allows us to incorporate additional treatments or treatment approaches into our experimental designs. This also allows us to randomize our treatments within an acrylic rack apparatus for maximum robustness and being able to minimize potential block effects in experiments. That is a common thing to affect tank experiments. Now, there are more details of this study in a paper that was just released a couple of weeks ago, and I will share that link at the end of this talk. I'm also happy to answer questions. But what we found is that disease-inoculated sediments were able to transmit the disease to healthy corals in as little as 24 hours. So we're talking extremely rapid transmission of disease. When we compared the rates of disease transmission from sediments to, say, controls where there was direct contact between diseased and healthy coral, we found that there was lower disease transmission. But this is to be expected when you're talking about indirect contact between your disease source and your healthy coral. Using tissue histology as well as some of our visual observation methods, we found that the signs of disease infection were similar among modes of transmission. So between direct contact with diseased corals and with exposure to disease-inoculated sediments. So we're very confident that this is actually the disease manifesting itself. We also conducted microbial community profiling using the 16S ribosomal gene of sediment samples and found 16 bacterial genera that were more abundant in disease-associated sediments compared to control sediments. Some of these include known SCTLD indicator taxa like Vibrios and Rhodobacterales, but we also found some others that were a little bit more unique. And so at this point, we are unsure whether these represent opportunistic taxa that are just responding to the increased levels of necrotic tissue from decaying corals, or if they actually represent some type of pathogen or perhaps a pathogenic consortium. So we're hoping to conduct some more work in the future and look at more sample sets to see if these microbial trends exist across the board and in wild environments. There we go. From this study, we determined that the reef sediments can in fact serve as a source of SCTLD. And additionally, we found evidence of reservoir potential of these disease microbes and sediments. So this could potentially mean that disease-inoculated sediments might hold on to some microbial communities and allow them to even proliferate in some environments. From this, we can conclude that sediments likely contributed to the spread of SCTLD throughout Florida and likely also pose a continued threat to the persistence of the outbreak both within Florida, but perhaps also within some of the other territories that have seen recent observations of disease. From here, we cannot stress enough that coastal management priorities and disease mitigation strategies should be considering the role of sediments in the spread and persistence of the disease, in addition to, say, the movement of water among reefs. Additionally, efforts should consider potential treatment approaches to reduce transmission risk, and we are hoping to undertake some of these experiments to evaluate different treatments of disease-inoculated sediments. Expanding out to, say, the Caribbean level, one of the big priorities is to understand how the disease has spread to geographically isolated reefs within the Caribbean. And so one of the suspected modes of transmission is ship's ballast water. And so I will jump into that right now. There's been a couple recent studies that have hypothesized that ballast water transfer, or the movement of water in and out of ships' ballast tanks in order to maintain stability among ports, that they might be contributing to the spread of SCTLD among the Caribbean. So if we take a look at the map over to the right, each of the pink dots represent a commercial port, and the larger grayscale dots correspond to observations of SCTLD, with the lighter colored dots corresponding to earlier, so closer to the initial outbreak, and the darker dots corresponding to later or more recent observations. And as we see, when we get further down into the Caribbean islands, we start to see these more recent observations. Now, a lot of these areas are geographically isolated. There aren't many I'm sorry, my Siri just popped up. There aren't many oceanographic currents that might connect reefs in Florida with these reefs. And so this one study led by Rosenau found that there are some interesting comparisons to be made between ballast water transfers and then subsequent SCTLD observations. So they hypothesized that ballast water might be contributing to that spread. There are several regulatory agencies that already manage the treatment and release of ballast water, as well as in the management of microbial testing standards to make sure that all ships are following the regulations and have functioning treatment systems on board. And so we sought out to test whether ballast water testing and treatment standards are effective in mitigating SCTLD. So this would require really no additional action in combating the spread of the disease from ships. So we conducted 2 experiments here at AOML. The first was testing whether UV treatment of disease-inoculated water was effective in reducing transmission risk. And secondly, we had addressed whether SCTLD can persist in ballasted water from sealed containers to simulate a ship, say, moving across the Caribbean. The beauty of our system at AOML is that it's fully modular, and so we can incorporate inline treatment approaches like this UV chamber shown to the right that allows us to generate both untreated disease-inoculated water as well as UV-treated disease-inoculated water. Then this water is passed on to those individual coral vessels that I described earlier, and we can look at transmission dynamics. From this experiment, we found that both disease-inoculated water and simulated ballast water were able to transmit SCTLD, once again without direct contact between diseased and healthy corals. In calculating the amount of water needed to initiate infection, we found it was relatively large, on the order of about 10,000 liters. But once corals showed their initial signs of infection, the lesions progressed rapidly across the coral fragment. In a lot of cases, we'd have a healthy coral one day, and then the next day we'd come in and we'd have a completely bare skeleton, as indicated in that top right photo. Additionally, we found limited evidence of a pathogen concentration effect when we ballasted water. So in some treatments, we found that ballasting was required to initiate infection in downstream corals. So this would suggest that over time in a sealed environment, the SCTLD pathogens might even be proliferating. Additionally, we found that UV treatment of disease-inoculated water reduced transmission rates by 50% for the 2 species that we examined. But interestingly, it did not have an effect on the volume of water needed to initiate infection. So fewer corals got infected, but it still happened in about the same amount of time or amount of water distributed to the corals. So we're not sure why this is happening, but I think we need to do some more work into this and specifically use some omics technologies so that we can look at the composition and abundance of microbes in these disease-exposed water, as well as in our treated water. From this experiment, it seems clear that ballast water is posing as a threat to SCTLD transmission among the Caribbean, but I think there's some silver lining here. We found some evidence that UV treatment was effective and may therefore mitigate transmission risk. And this is possibly through reduction of pathogen load. And so we're hoping to look at microbial abundance, as I mentioned in the previous slide, to see if there really is an effect on the microbial communities as they pass through that UV sterilizer. Second, we found some evidence that the SCTLD might be persisting in ballast water systems and perhaps even proliferating. So this is an avenue that definitely needs to be looked into, and we're hoping to design some more experiments at perhaps larger scales to address whether ballasting has this effect. From here, there are a couple critical next steps. The first is that we need to evaluate multiple ballast treatments. So ballast water treatment systems are often more complicated than the system that we tested in our lab. They will combine treatments. So they'll have an initial filtration, and then they'll combine it with some type of biocide like UV, like we tested, or perhaps a combination of chlorination or ozonation to neutralize any kind of microbial communities. Second, we need to be conducting risk assessments to determine the relative risk of vessel discharges or of transmission from ports throughout the Caribbean. So this is an activity that NOAA is very well aware of and is identifying some teams that can be able to do this on a large scale. And finally, from some of the microbial data that we'll be generating from this study, as well as from some field data that we are suggesting needs to be collected, we need to develop an SCTLD diagnostic for ballast water systems. In a perfect world, I think of an indicator test kit, just like we have our rapid COVID test that you dip into a ballast water tank and it tells you, yes, this water has been exposed to SCTLD, or no, it hasn't. This would be the ultimate goal and would allow us to rapidly assess the spread of disease throughout the Caribbean. Now, finally, I'm going to take a bit of a left turn here. And while the previous 2 studies have been focusing on SCTLD transmission, now we're going to focus a little bit more on how we're responding to the coral disease outbreak at the colony level. And so I'll be focusing on a disease intervention project that I was a part of using antibiotics. Now, there are several teams throughout Florida that have been treating now thousands of diseased corals since 2019, and they're using an antibiotic with a specially designed paste meant to release that antibiotic over a period of 2 weeks. Now, while these efforts have shown that generally we have cessation of lesion progression in the treated colonies, we have a very limited knowledge of potential negative effects of this antibiotic treatment on corals and their symbionts. And so we conducted 2 different experiments with transcriptomic analysis. The first being an ex situ transmission experiment to see what genes are differentially expressed following exposure to SCTLD. And the second is an in situ intervention experiment where we looked at changing gene expression patterns in response to antibiotic treatment. I'll be presenting the results of our ex situ transmission experiment first, and right off the bat, we can tell that there are massive transcriptomic differences between diseased and healthy individuals on the order of about 7,500 differentially expressed genes. What's interesting here is that some of the corals that were exposed to the disease but did not end up showing visible disease signs, these we call no active infection corals, they were more similar to their healthy controls than they were to the other diseased corals. So this indicates that there might be some potential resistance in some of these coral genotypes, but this requires further testing. When we look at our field data, right off the bat, you may notice that it's a lot messier, and there's generally a lot of variability in transcriptomic data sets from field samples because of the environmental variability. But what we're able to do is look at pairwise differences between treatments. So we can compare healthy corals to diseased corals to treated corals. And what we found after 2 weeks following antibiotic application, the treated corals more resembled their healthy controls than they did their original disease state. So this is suggesting that the antibiotic application is a bit of a reset button for these corals, that they are once again maintaining some level of healthy homeostasis versus their original disease state. From these 2 different experiments, we can come to a couple conclusions. The first is that SCTLD infection is causing large-scale depression of some key processes, physiological processes, such as metabolic and repair mechanisms are generally downregulated, as indicated in the figure to the right, some of these darker blue squares. We also found some evidence that some coral genotypes might be resistant to SCTLD, and we don't know whether this is fully due to the coral's genotypes or their symbiotic communities, or maybe even a combination of both. This obviously requires some further testing that we're hoping to get into. In general, it seems that intervention efforts using antibiotics are effective in stopping the progression of disease across a coral colony. And from our transcriptomic data, it would suggest that it doesn't have an adverse effect on coral transcription through time. So antibiotic treatment might be warranted for certain cases where you're trying to keep extremely rare species like this Dendrogyra cylindris alive, or if you're trying to maintain a healthy, reproductively viable population by treating some of our larger coral colonies that we have in Florida. Some of our ongoing work as part of a collaboration with Mote Marine Lab and is whether or not we can identify gene biomarkers that are indicative of SCTLD resistance. And so right now we're conducting transmission experiments over at Mote in Sarasota, Florida. And downstream we'll be looking at the coral genotypes, transcriptomics, so gene expression in response to disease challenges, and finally microbial and symbiotic communities to see if we can identify some key differences among corals that get infected versus corals that tend to be more resistant. So I'm really excited for this project, and I'm hoping that this will allow us to generate some real actionable tools to start screening wild populations. So with that, I know it's a lot of information, but I really appreciate everybody's time. I'm happy to answer some questions. Likewise, my email address is posted up here on the screen. And finally, in the bottom right corner, there is the citation for one of our recent papers addressing sediment transmission with this disease. So if you are more interested in that project, feel free to check it out. Thank you all. Happy to take some questions.
Excellent. Thank you so much, Michael. That was a fantastic presentation. We do have a bunch of questions that are coming in, so I'm going to go ahead and start from the top. This question comes from Megan. Do we know to what degree ships currently treat ballast water? Is this something that is consistently done, or is this something that slides under the radar more often than not?
That's a great question. So there are very tight standards for how ships treat their ballast water. But I will say there's a lot of ships out there and there's a lot of ports. And I think ballast water management, without trying to throw anyone under the bus here, ballast water management is very closely regarded in territories like the United States, but perhaps is less so throughout other areas of the Caribbean. There's also possibility that other parts of the ship might be contributing to the spread of disease. Namely the biofilms that tend to accumulate on the outside of the ship hull. There's really no regulation for how biofilms are managed, and very few ways to actually treat that. And so it's likely that ships by themselves might be serving as a source. And so it could be a combination of ballast water systems as well as hull biofilms.
Excellent, thank you. All right, we have a couple other questions here. This one is from Victoria. Are there concerns regarding antibiotic resistance leading to worsening cases of SCTLD?
Yes, absolutely. And this is why antibiotic treatments have really been tested in Florida. There are other regions throughout the Caribbean that are hesitant to do this. The reason why it was approved for use in Florida is because through comprehensive water testing of coastal regions, we found, and I say the royal we here, we found high levels of antibiotics already in the water. And so the justification for using amoxicillin for coral treatments was that it's already present within these reef environments. And so if there's going to be any kind of antibiotic resistance, it could possibly already be in effect. And so some of the concerns with using this treatment throughout other areas of the Caribbean is that a lot of these smaller Caribbean territories don't necessarily have high concentrations of antibiotics in their coastal waters. And so it'd be a relatively new chemical treatment being applied in those areas. So yes, there is concern for antibiotic resistance. I would also say that we have very little knowledge about what that would mean for corals, and very few ways to test it. I mean, this is something that has been championed for human health, but in the coral medical field, our capabilities are far more limited, and we need to do more work into immune responses in coral populations. Great question.
All right, thanks, Michael, and thanks for the question, Victoria. Our next question comes from Courtney. Great presentation. Is there a minimum concentration of pathogenic microbes in sediment or water that causes SCTLD in ESA species?
We do not know yet. It's something that we'd like to test. There are a couple downstream experiments that we started to think about immediately after seeing the results of both our sediment and ballast water studies. The first is we don't know how long these inoculated sources remain infectious. And so we are hoping to conduct some time series experiments as well as in some dosing experiments to see if we can figure out essentially how much pathogen is needed to successfully cause disease signs in corals. Now, I will say in the sediment experiment, in some of our disease treatments, we're working with extremely small areas of coral tissue, diseased coral tissue, in order to generate that sediment. We had a 5 square centimeter coral fragment, so that's about an inch by inch square. And that was apparently able to generate enough pathogen that was shed to the sediments and then able to transmit it onto a healthy coral. So I suspect it's probably very, very little initial pathogen is required. And then if sediments can in fact serve as a reservoir, it's possible that they might even proliferate in that type of environment.
Excellent, thank you. And we have one last question here from Admiral Gallaudet. Excellent presentation, Michael. I love the development of NOAA's agency-wide strategy to prevent SCTLD from spreading to the Pacific. Do you know if it has remained outside of the Pacific?
As far as I am aware, it has not been transferred yet to the Pacific, but there are several groups, research groups, including our lab here at AOML, that are very interested in conducting some transmission experiments with Pacific species. We feel that this is one of the highest priorities, and as you mentioned, is one of NOAA's highest priorities in responding to the disease outbreak. And so we are hoping to rapidly mobilize some of these transmission experiments so that we can understand first whether Pacific species are even susceptible to the disease. And second, we're hoping to mobilize some monitoring efforts in some areas of the Pacific, such as Panama, that would be one of the first zones likely to see disease observations if it does spread. So we're hoping to increase our monitoring efforts in those areas so that if it does spread, we can catch it quickly and we can immediately begin responding. And I say the we here, but really this is an effort of many institutions. NOAA is particularly championing this effort. And so we're working with other research groups, other regulatory partners to ensure that the research that we conduct here at AOML not only meets these management needs, but gives the information, the critical information needed to management partners in these different areas so that they can act on it in the easiest way possible.
Excellent. Thank you so much again, Michael. If folks have any other questions, I encourage you to continue to ask them in the chat and I will forward them to Michael, or you can reach out to him directly. His email is up here on the screen. I think I can say on behalf of everyone that we're really looking forward to having you back at the Omics Seminar Series next year to present on some more of these next steps.
Thank you. Yeah, I'm really excited to see where a lot of the subsequent work from these projects comes out. We've got some interesting omics pipelines planned for some of these data sets we've collected. And I think, however depressing, I think that there's a lot of little glimmers of hope that are coming out of the research that we're doing here. So thank you, everyone.
Yeah. All right. All right, so we have our next presenter. So Anna, I'm going to go ahead and give you screen sharing permission right now. And while you are getting your presentation situated, I'm going to go ahead and present who you are. So thank you so much again, Michael. Really appreciate it. Our next presentation is an omics approach to multi-stressor resilience in the threatened coral Acropora cervicornis, presented by Dr. Anna Palacio. So Dr. Anna Palacio is an NRC postdoctoral research fellow within the AOML Coral Program. Her research has focused on the effects of ocean warming, acidification, and pollution on corals, with emphasis on the coral-algal symbiosis and bleaching. Her goal is to help to identify environmental and genetic factors that promote more resistant corals and allow them to survive in our fast-changing world. Anna, thank you so much for presenting today. I will now pass it off to you.
Thanks, Catherine, for the introduction. Can you hear me okay?
Yep, we can hear you great.
Awesome. So yeah, it's great to be here today and share some of the research I have been doing at AOML to better understand the factors that promote resilience in the threatened coral Acropora cervicornis and how we can use omics tools to understand the mechanisms behind this resilience. First of all, I would like to start thanking all the people that have been part of this research, including the coral nurseries and restoration partners that have donated corals for the studies, my mentors, advisors, and collaborators, the undergrad and grad students that helped to run the experiments, and all my lab mates at the AOML Coral Lab and the University of Miami Coral Reef Lab. So to put in context the importance of understanding Acropora cervicornis resilience, we need to look at the trends of the Caribbean coral reefs. Caribbean reefs have been exposed to multiple anthropogenic and natural stressors for decades, and these stressors have caused the loss of 50 to 80% of the coral cover since the 1970s. A significant proportion of this coral cover loss is explained by the massive reduction of the once abundant corals, Acropora cervicornis and Acropora palmata. This species used to be dominant reef builders in the Caribbean. However, their population started declining dramatically in the 1970s. About 80 to 90% of the Acropora cover was lost from the 70s to the 2000s. And as a result, both species are now listed as threatened under the U.S. Endangered Species Act. The main causes that were reported for Acropora's decline are disease, especially white band disease, but also coral bleaching and reduced water quality as a result of growing human populations in coastal environments. So protecting and restoring Acropora cervicornis populations is a NOAA priority, not only because this is an endangered species, but also because acroporids are key for Caribbean and Atlantic reefs. These corals grow up to 5 to 10 times faster and taller than our Caribbean coral species, and therefore they contribute more to the reef architectural complexity and carbonate production than other coral species in the region. So NOAA's restoration work concentrates on 4 strategies, and in the frame of that work, my own research has focused on understanding coral resilience to individual and combined stressors, so we can use that information to improve coral survivorship and to increase restoration success. We can look at both environmental and genetic factors that can shape coral resilience and use that information to optimize management and restoration at a local scale. So when we look at the environment, we see that corals are still facing multiple stressors, and these stressors often occur at the same time. And some of those stressors then could interact and result in synergistic effects that are more detrimental for the coral reefs than the impacts of the individual stressors. But also we know that some stressors are easier to manage at a local level than others. So a key question is how important is to reduce those local stressors such as nutrient pollution to increase resilience to other factors that are harder to manage, such as warming and disease. Also, considering that some of the stressors will be part of the reef environment, at least for the next decades, a second question is, can we identify coral genotypes that are more resistant to these stressors? And then should we target these genotypes to restore specific sites? However, the picture is a little bit more complicated because corals are meta-organisms that are composed by multiple parts. The corals live in association with a diverse community of symbiotic algae in the family Symbiodinaceae, but also bacteria, archaea, and other microbes that share resources among them and recycle nutrients. So when we are looking at a coral response to stressors, we're most likely looking at the response of the different parts of the whole biome. So to start answering these questions, how can we improve resilience and can we find resistant Acropora cervicornis genotypes? We performed 2 experiments in which we tested the effect of pre-exposure to elevated nutrients on different genotypes and how that pre-exposure to nutrients determined response to heat stress in experiment 1 and then disease exposure in experiment 2. So in experiment 1, we tested 2 elevated nutrient treatments, ammonium only and ammonium plus phosphates. However, the addition of phosphates did not change the effects of elevated nutrients or ammonium alone. So in the second experiment, we only tested elevated ammonium. After the nutrient exposure in experiment 1, we gradually increased the temperature for all the nutrient treatments and compared the response to heat stress among genotypes and nutrient treatments. And in experiment 2, we exposed half of the corals in each nutrient treatment to either a placebo or disease homogenate. So to do that really quickly, we collected healthy and diseased fragments from the field, and then we blasted their tissue to produce either placebo or disease homogenates that were later used to expose the corals in the nutrient treatments. So through both experiments, we monitor the effect of the stressors on each coral genotype performance, also their algal community composition and function, and the prokaryotic community composition. So briefly, we monitor survivorship of the corals every day. Every week, we measure the buoyant weight of the corals to estimate the growth rates. And every week, we also monitor the photochemical efficiency of the algal symbionts. This metric, the photochemical efficiency, is also called FB/FM. And pretty much all we need to know is that it's one of the most common metrics to assess stress in photosynthetic organisms. And finally, we used DNA-based estimations of the composition of the algal communities and prokaryotic communities. So for the algal symbionts, the families in Butynaceae, we use qPCR assays that pretty much quantify the amount of DNA that come from a specific symbiont types versus how much DNA is coming from corals. So this gives us an estimation of symbiont abundance called the symbiont to host cell ratio, which is pretty much how many cells of a specific symbiont we have per each coral cell. But spoiler alert, all the corals hosted the same symbiont species, and the abundance of the communities did not change under the nutrient treatment. So I'm not going to show further results for this. For the prokaryotic community, we use amplicon sequencing of the 16S ribosomal gene. And through bioinformatic pipelines, we determine the composition and relative abundance of the bacteria. I'm gonna jump into the results of experiment number 1. So here I am presenting the effects of pre-exposure to elevated nutrients and followed by heat stress. So in all of these 3 metrics, coral survivorship, coral growth, and photochemical efficiency of the symbionts, big numbers are good, lower numbers are bad. We have in the colors the different nutrient treatments. So blue is ambient, green is ammonium, red is ammonium plus phosphate. So we can see, and in the frames, the red frames, I am highlighting the time in which the corals were exposed to high temperature. So what we can see is that elevated nutrients alone reduce the performance of the coral host. It caused some mortality as well as reduction of the growth rates. However, those effects exacerbated once the coral were exposed to high temperature, and mortality really accelerated during that time, and also the growth declined a lot faster. The effects on the symbiont community were a little bit different. Initially, under controlled temperatures, elevated nutrients increased the photochemical efficiency of the algal symbionts. Then when the corals were exposed to heat stress, they experienced faster and stronger declines in the photochemical efficiency. We also wanted to look at the variation of these responses by coral genotype. So here I'm showing again coral survivorship and coral growth. I am only showing the effects on the corals that were exposed to elevated nutrients. And in this case, each color is one genotype, and the color is matched between the survivorship and the growth rates. So what we found is different genotypes have different susceptibilities to elevated nutrients, and elevated nutrients followed by heat stress, with some corals experiencing mortality along before than more resistant genotypes. And those patterns are similar in terms of the growth reduction. Some coral genotypes were able to maintain higher growth rates when they were exposed to elevated nutrients and heat stress. So that is really interesting because it tells us that we could use some of these more resistant genotypes to restore reefs that could be exposed to elevated nutrients. But we also wanted to understand, like, where is this resilience coming from? And to do that, we look at the composition of the microbial community to see if they could be part of this differential response. So here we have in the left panel the corals before they were exposed to heat stress, and here we have on the top panels the ambient treatment, and then center the elevated ammonium, and then in the bottom the corals in elevated ammonium and phosphates. So what we found is most of the corals were dominated by one bacteria taxa in family Meteorological. And when the corals were exposed to elevated nutrients, that only caused very modest changes in the microbial community. The microbial community composition was kind of stable when the corals were exposed to high nutrients. However, when the corals were exposed to first 1 week of heat stress, but even more 2 weeks of heat stress, that stress did cause significant changes in the microbial community with an increase of the Alteromonas especially. But not only that, the bacterial community changed differently in the corals that were exposed to elevated nutrients. We also look at the bacteria that had differential abundance among different genotypes and correlated this bacteria abundance with the survivorship of the genotypes under elevated nutrients and heat stress. And we found that 2 bacteria taxa relate with higher survivorship rates in these corals. So we think that these 2 taxa could be used to identify other potential nutrient resilient genotypes for restoration. So I'm gonna switch gears here to experiment number 2. Remember, in this experiment, we tested the effects of elevated nutrients, and we exposed the corals to disease. So here we have 10 genotypes. So each genotype is in a different panel on this figure. And then in the colors, we have the different combination of treatments. So different nutrient treatment with homogeneous treatment. Again, we saw a huge variation in the response of these different genotypes to stressors. We have one genotype that was only showing mortality when it was exposed to both elevated nutrients and the pathogen, but did not show mortality when it was exposed to the pathogen alone or elevated nutrients alone. We also have some genotypes that experience mortality when they were exposed to the pathogen, regardless of the nutrient treatment. We have more susceptible genotypes that started experiencing mortality when they were exposed to elevated nutrients alone. So even before they were exposed to the homogenates, some of these corals started dying in the elevated nutrient treatments. Here we're going to look at the preliminary results from the microbial community of these corals to try to understand some of the survivorship patterns. And on the top, we have the corals that were exposed to ambient nutrients and the placebo homogenate. And then in the bottom, we have the corals that were exposed to elevated nutrients. So again, similar to our first experiment, nutrients alone did not cause a lot of changes in the composition of the microbial community, with the exception of one genotype in which there were more significant changes. However, when we expose these corals to the pathogen homogenates, we identify higher relative abundances of some taxa, including Alteromonas, Flavobacteriales, Oceanographic, and Vibrios. And interesting, when we look at the microbial community of the homogenates that we use in the experiment, we found that this differentially abundant taxa look very similar between the pathogen homogenates and the new bacteria detected in the corals that were exposed to these pathogens. So identifying coral pathogens, it's really hard. But we are excited that we have some clues that could lead us to continue testing this specific taxa as potential pathogens for Acropora cervicornis. So to summarize the results of these 2 experiments, we found that elevated nutrients alone is enough to reduce Acropora cervicornis survivorship and growth. And not only that, but pre-exposure to elevated nutrients also exacerbate the effect of heat stress. While the pre-exposure to elevated nutrients only have additive effects when the corals are exposed to disease. We also found very strong variation in the susceptibility of different genotypes to the individual and combined stressors, and that includes nutrients, heat, disease, and the combination of this. And also we found evidence that nutrient-resilient genotypes could be identified by their association with a specific bacteria. Finally, from our last experiment, we found some bacteria taxa that could cause disease in Acropora cervicornis, although we need more testing for this. So why do we care about all of this, or what do this mean for management? So first, based on these results, we now know that improving water quality is key to restore and maintain Acropora cervicornis populations. When we tested the effects of elevated nutrients in Acropora cervicornis, we ran this experiment with other species. And the response of Acropora cervicornis to elevated nutrients was a lot stronger than other corals, such as Orbicella fragiolata and Statisteria. So nutrients really matter for Acropora. And therefore, when we are considering the location for our Acropora nurseries or outplant sites, we really need to consider water quality if we want to improve survivorship. Also, we could increase restoration success by prioritizing these nutrient-resilient genotypes to restore reefs that are more likely to be exposed to elevated nutrients, such as in inshore locations. And also we could allocate more nutrient-susceptible genotypes to sites with lower risk of elevated nutrients, such as offshore locations. Also, we could develop biomarkers for the bacterial taxa that would allow us to identify more nutrient-resilient genotypes without having to run them through this stress experiments. And finally, perhaps we could identify some of the disease-causing agents for Acropora cervicornis. That's all I have. I'm gonna take some questions next. I hope I'm not over the time a lot.
Thank you so much, Anna. You are not over the time, no worries.
Awesome.
That was a fantastic presentation. We're getting a couple of questions coming in, so folks feel free to continue to add them in here. Okay, so Anna, I actually have a question for you. Looking at the experiments and the results from your experiments, it seems like some of the genotypes that you're looking at do eventually, you know, like their health declines over time still after like the persistent exposure to nutrients and heat. Is that just something that's happening in like an experiment setting, or do you expect something like that to happen out in the field, or is it like a more fluid environment in the field, so we expect these more resistant genotypes to survive out in the wild?
So that's a really interesting question. I guess one, like a key decision when we run this experiment is the level of nutrient stress that we apply to the corals and what is realistic, right? So initially we use a target that kind of double what we see in polluted reefs, and we were like, okay, maybe this is not realistic, and we're finding effects that perhaps are not gonna mirror what's gonna happen in the field. But unfortunately, when we were running these experiments and collecting water samples from our ambient tanks, we were able to detect very high nutrient levels happening naturally in the water from Biscayne Bay. So I think our results are definitely applicable for the field, and in these reefs that are experiencing high nutrient levels, we could see this reduced performance, including reduced growth rates and survivorship. But I think even with that, a key part of this is when these reefs are exposed to heat stress than during the summer, then we're going to see a stronger effect of the nutrient pollution. So I guess my short answer is yes. I think we could see those decline performance in the field as well.
Okay, awesome. Thank you so much. But I can totally see the value of having those resistant genotypes be out there still, so they have a shot at surviving a little bit longer.
They buy you extra time. So if you have not such a strong nutrient inputs, you increases the chances that they're gonna survive, right? So we have some survivors to the end of the experiment, and some of those resistant genotypes lost a few of the fragments, but some of them made it to the end of the experiment. So if we're looking at the reef and we could save some of the corals by choosing resistant genotypes, I think that's good.
Excellent. Thank you so much for answering my question. We have one that came in from Katie. She asks, great presentation. Where did these A. cervicornis genotypes originate from? Where were they collected?
They were collected from the Key Biscayne nursery by the University of Miami restoration team.
Awesome, thank you. All right, and we have a final question here. I think this question could probably go to both Michael and Anna. So Michael, I hope you're still on the line. This one's from Admiral Gallaudet. He says, when he was working on the omics strategic plan with Kelly Goodwin, he learned how important machine learning was in omics data processing. Do either of you want to describe what that process is like for the audience?
Oh boy. I will admit I have very little experience with machine learning approaches, but the applications that I see for a lot of the coral disease work that we're doing and that NOAA is doing in general would be to take the information that we're gaining about putative pathogens, or at least members of pathogenic consortia, and feed those into these machine learning models that create these simulations of probability. So it would be to identify and confirm potential pathogens within our sequencing data sets. There are other approaches as well from Anna's perspective in assessing multi-stressor predictions of coral reef health, where you'd be able to feed in molecular data sets like transcriptomic data sets or proteomic data sets and make predictions from the relative expression levels of those genes or proteins respectively, to then predict whether a genotype would be able to survive in a stressful environment. That's about the extent of my knowledge with it, but we are hoping to increase our ability to integrate artificial intelligence and machine learning approaches into our molecular pipeline so that we have better inferential power when it comes to making predictions of coral colony health or coral reef health at large.
Thank you, Michael. And also we had a talk that as part of this seminar series that touched on this exact topic. So I'll put that in the chat now. That was Dr. Anderson Mayfield's presentation, who's also a researcher at AOML. So I encourage you to please check out that talk.
Absolutely. He has a lot more information in that than I was able to provide.
Yeah, he's very knowledgeable about that. Well, Anna, thank you so much for your presentation. And Michael, thank you again for presenting as well. And I also want to just take a moment to thank the entire audience for tuning in today. We really appreciate it. I have to give you all my quick spiel as we're wrapping up here. Normally, the NOAA Omics Seminar Series takes place on the third Wednesday of every month at 12 p.m. Eastern Time. However, our next seminar is actually going to take place on the fourth Wednesday in February due to a scheduling conflict of my own. So February 23rd, 2022 at 12 p.m. Eastern Time. Our speaker is Wes Larson from the NOAA Alaska Fisheries Science Center. So registration for that talk and more information about it is currently up on our NOAA Omics website. And with that, I want to thank our speakers and our audience again, and I will go ahead and sign off now.
Thanks, Catherine, and thank you everyone for attending.